Okay, before we start, just an announcement. I have some good news and some bad news. And so we'll always start with the bad news first. So the bad news is that there will be no lecture next Monday, September 19th. So that's bad news. But there's good news. Wait. So don't come next Monday. I'll be out of town until Tuesday night. So the good news, there is good news. Monday's lecture will be a screencast. So you're not going to really miss a lecture. You're going to have to watch it online as a screencast. You just don't get to watch me give the lecture. So maybe that's the good news. Okay. So we left off with my little teaser trying to tell you that I was going to reveal to you my theory on how it was that microbes facilitated the change in human community structure known as the transition between the Paleolithic and the Neolithic period. So remember that during the Paleolithic period, our ancestors are all hunters and gatherers. They're moving around, they're following herds, they're following native crops as they might ripen or be ready for harvest. And then eventually we move into this more sedentary, agrarian culture that we still live in today, where we have domesticated animals and we grow our own crops. There's less hunter. There still are some people, fortunately, that still adhere to a hunter-gatherer lifestyle, but for the most part, most of us are now in this agrarian Neolithic. So there are thoughts that part of what caused people to begin to kind of slow down the hunter-gatherer lifestyle had to do with crops, native crops, wild crops, that were particularly valued, partly because of their energy production, but partly because of their taste, their sweetness. Things like fruits and berries. So these are things that you can imagine, since we all still love them today, that as a hunter-gatherer group or extended family group moving around, that when you found the area with like lots of wild raspberries or wild grapes or things like that, those were a major resource and cause for celebration. So another thing that happens with the consumption of things like wild grapes and wild berries is that as they spoil, what do? Microbes, by fermentation, make what we now know today as beer and wine. But of course, back in the day, imagine, caveman here, sitting around the campfire having just consumed some naturally fermented grapes and having a little buzz on. So this is a feeling that we still many of us, crave today. So there's a feeling that what may have begun as just a good time, eating these fermented products and telling great stories around the campfire, because you all know that as the more you drink, that mammoth got even bigger, right? That mastodon was even faster. So all of these things probably led to oral folklore, but probably because people began to value these crops so much, they started to protect them. They started to keep other people away from them. Weeds started to grow in the berry patch or around the grapes. They'd pull them out, the beginnings of cultivation. So possibly this microbial fermentation that leads to things that many of us still enjoy today, were actually part of what caused us to settle down from hunter-gatherers. And that's all microbial fermentation that's done that. So my hypothesis is that microbes have led to the current society that we have today. And of course, that microbial revolution that led to alcohol has led to very unintended consequences, such as things like this, and the bro culture of fraternities on campus. So we can blame bacteria and microbes for certain things because this is what we have today. Okay, so now everyone is really fired up to talk about microbial physiology, fermentation, because now you know how important it is. So really, if you think about it, we've talked a little bit about this already. You know, all life forms on Earth use either sunlight or chemical energy. And, you know, you and I fall in here in the chemoorganotrophic area. And remember that the chemolithotrophy, this is the type of ecosystem that I talked about with the deep sea hydrothermal vents, where sunlight plays no role, there's no photosynthesis, and inorganic molecules are being oxidized. So things like hydrogen sulfide, H2S. So these three forms of energy, chemical energy, are all used to do work. And as we go into our discussion of physiology, and I know that this is going to be very redundant for probably all of you, many of you anyway, I just want to make sure that everyone's kind of on the same page to go over a few of the definitions of the terms that we'll use a fair amount in here. So we're going to talk a lot about oxidation and reduction reactions. And so just so people remember that when something is oxidized, electrons are removed from it. And also important to remember is that in nature, electrons do not exist in isolation. You can't just store an electron in water. It's always going to be associated with something. So when a molecule becomes oxidized, some other molecule must be reduced because there has to be a carrier for that electron. So the molecule that holds on to that electron removed by oxidation is reduced. So it has gained electrons. And the nice thing about the biological systems is that when you see these systems, there's usually an extra H attached to the name. A proton has usually been associated with that reduced molecule to balance the electrical charge. So you can often tell the difference between an oxidized form and a reduced form. Probably the best example that we're going to talk about a lot is an electron carrier called NAD, nicotinamide adenine dinucleotide. When you see it as NAD+, that is the oxidized form. When it becomes reduced, when it gains an electron, it also gains a proton. So it's NADH. So that's the reduced form. That has an electron associated with it. So in terms of recognizing oxidation and reduction, it's really pretty easy to do. And I'll show you this in more specific examples. We're going to talk a lot about one of my favorite topics in microbiology, as you can tell from my appealing to your love of alcohol, fermentation. So fermentation is something that, while you and I do it slightly, you know, we have the capability of doing fermentation within our own cells, it's pretty restricted. Fermentation, especially in multicellular creatures like you and I, is pretty unusual. But in bacteria, it's oftentimes the only game in town. So there are a large number of bacteria, such as the streptococci, excuse me, that are in your mouths and throats right now, and other places as well. These are microbes that can't do respiration the way that you and I do. They rely entirely on fermentation. So in fermentation, these oxidation reduction reactions that go on, you know, where foodstuffs, carbohydrates and such are being oxidized, those electrons end up going on to some kind of an internal electron acceptor. This is what happens in the absence of an external electron acceptor. The best way to think about this is to think about what's going on in you and I right now as we breathe in oxygen, and oxygen is transported to our cells via the bloodstream. Oxygen is the final electron acceptor that you and I use. So the electrons that are removed by oxidation from the foodstuffs that we're digesting end up ultimately on oxygen, producing water. When you reduce oxygen, you generate water. That's an external electron acceptor because you breathed it in from the atmosphere. That's the opposite of fermentation. In fermentation, that final resting place for the electron comes from the substrate being oxidized. Hello? I get a free cruise to the Bahamas. That's really cool. I'm serious, that's what it was. It was a recorded call saying I'd been randomly selected and I won a free cruise. So that's where I'm really going on Monday. The hell with going. Who wants to go to Texas? I'm going on a cruise. Okay. I was afraid that something was happening on campus. I would normally have ignored that. Anyway, so fermentation is where the electron's final resting place comes or is part of the food being oxidized. We'll talk about the specific examples of that. In fermentation, that terminal electron acceptor, as I'm just mentioning now, is part of the substrate being oxidized. And in fermentation, basically the only way, or perhaps it is the only way, that energy is generated, or ATP is generated, by something known as substrate-level phosphorylation. In essence, that means glycolysis. It can also mean the TCA cycle. But I'll show you specifically where the ATP, the phosphate that's being put on, this third phosphate, is actually coming from the substrate being phosphorylated. It's one way to produce energy. It's the least efficient way to produce energy. An organism that is generating all of its energy by substrate-level phosphorylation is not very efficient. Respiration, on the other hand, respiration is what you and I do, and it's what many bacteria do. It's a much more efficient system. In respiration, there still needs to be an external electron acceptor, or there still needs to be an electron acceptor. In this case, it's coming from outside. It's not part of the food being oxidized. You and I are using oxygen. And as it turns out, there are, in vertebrate cells, in eukaryotic organisms, the only possible electron acceptor is oxygen. And it is the single most efficient electron acceptor there is. You can get no more energy from the oxidation of anything by putting those electrons on any other molecule than oxygen. So our respiration system is the most efficient. Bacteria also use our same, or many bacteria use our same respiration system. So the best example that we all know about is oxygen. But this is where bacteria are, and we'll go over this in some detail, really a lot more metabolically diverse and better able to adapt to many environments than you and I. You and I have the ability to use this highly efficient oxygen as a terminal electron acceptor, and so do bacteria. But bacteria, as I mentioned when we were talking about the dead zones in the Gulf of Mexico from phytoplankton blooms, bacteria can switch from using oxygen during respiration to something slightly less efficient, but still allowing life to continue. So many bacteria, these are bacteria that are referred to by the term facultative anaerobe. These are microbes that can switch from using oxygen as an electron acceptor to using something like nitrate or sulfate, reducing it to nitrite, and actually even eventually reducing it all the way back to atmospheric nitrogen, so it's given back off as gas. So you and I can't switch from doing this, but microbes have such great metabolic diversity and utility that oftentimes they can switch and do respiration, or what's known as anaerobic respiration. It seems like a bit of an oxymoron, because you and I think of respiration as something that we do to bring in oxygen. Anaerobic respiration just doesn't make sense. But by anaerobic respiration, we simply mean that something other than oxygen, something like nitrate, is being used as the terminal electron acceptor. And in respiration, the largest part of ATP is generated not by substrate level phosphorylation, not by things like glycolysis, not by things like the TCA cycle, but instead ATP is generated most efficiently and most abundantly by oxidative phosphorylation, which is the opposite, if you will, of substrate level phosphorylation. And that's done by electron transport chain. So these are just some terms that you can refer back to. You probably don't need to. That was probably a waste of my breath and your time. But to show you a little bit more graphically the difference between substrate level phosphorylation and oxidative phosphorylation, this cartoon actually illustrates it a little bit better. In substrate level phosphorylation, imagine a pathway that's like this. This is in essence glycolysis, or it could be the TCA cycle perhaps. But think of glycolysis. You'll notice that when ADP is converted to ATP at this step between C and D, the phosphate group that's being put on to ADP to make ATP comes from the substrate. So this makes sense why we call it substrate level phosphorylation. This occurs mostly in glycolysis. There is at least one step of substrate level phosphorylation in the TCA cycle. It doesn't generate ATP, but it generates GTP instead. So that's substrate level phosphorylation. It's very important, but it's not highly efficient. It's going on in your cells and my cells right now, but it's not really a major source of ATP inside our cells. Instead, what's more efficient, and what you and I are doing, and what many bacteria do, is oxidative, or electron transport system. That's what the ETS here stands for. More often referred to as oxidative phosphorylation. This is where bacteria use that proton motive force that we referred to a couple of times earlier in this class to generate electricity, to generate ATP. In essence, so we talked about the ability of bacteria to rotate their flagellum by allowing protons, which are at a higher concentration outside of the plasma membrane, to come back across the membrane and use that decreased potential energy. So the decreased battery charge here, a positive charge outside, negative charge inside. As each proton goes through, that potential energy is decreased and the flagellum rotates. Or in active transport, something is some type of a solute or nutrient is imported. So we've seen at least two different examples of how it is that this proton motive force can be used to do work. But one of the most important things that proton motive force does is to generate ATP by oxidative phosphorylation. And you've all studied this before, even if you haven't taken a microbiology course, in virtually any biology course when you've talked about mitochondria. And you should always remember that mitochondria are, at least used to be, free-living bacteria. And so it's actually bacteria that invented oxidative phosphorylation. And eukaryotic cells have simply co-opted bacteria and now allowed the bacteria to do this work for them. And so we'll cover this a little bit later in this section, where protons on the outside of the plasma membrane at a higher concentration are now gated back across the plasma membrane through this F1, F0 ATP synthase. You've all seen this large nanomachine. Large nanomachine, that's an oxymoron. This nanomachine that has the ability to generate torque. It rotates as protons go through here, very much like the flagellum. So there's a very strong similarity between this. But now that rotational energy, that torque that's generated as these protons go through, doesn't drive motility. Instead, it drives the synthesis of ATP on the cytoplasmic side. So this is oxidative phosphorylation. You generate phosphate by decreasing the potential energy across the membrane. So this is a much more efficient source of ATP, or chemical energy for the cells. Now, I kind of mistakenly refer to this section as metabolism. In reality, we're not truly going to talk about metabolism, because metabolism is a collective term which means both catabolism and anabolism. The only point really in this semester where we talk about anabolism we've already done. We talked about the peptidoglycan synthetic pathway. So how is peptidoglycan put together? That's an anabolic pathway. Things are being built up. For the most part, exclusively from this point forward in this metabolism, in quotes, section of the class, we're going to talk about catabolism, the other half of metabolism, the breakdown of foodstuffs, of molecules for energy and other things. It's not just energy. So really what we're going to talk about is catabolism. And catabolism really has three different stages. And we've talked a little bit about the first one. So stage one, there's no energy production. So the bacteria, the microbes get nothing out of stage one. Stage one occurs largely extracellularly. So outside of the body of the cell, you have the secretion of enzymes into the environment to break down polysaccharides or proteins or lipids into bite-sized morsels that can then be brought into the cell. And then inside the cell, in stage 2, you can have these simpler molecules broken down into molecules that feed into things like the TCA cycle, shown here at the bottom, but the glycolytic cycle here. So now you begin to get some modest amounts of energy in the second stage of catabolism. And then the most energy produced is this third stage here, where you have the complete oxidation of the carbon source. So the monosaccharides or the amino acids that are being brought in are being oxidized via the glycolytic pathway. And some ATP is generated by substrate level phosphorylation here, but it's a fairly modest energy gain. Once you come out of glycolysis, this pyruvate now feeding into the TCA or carboxylic acid cycle to be completely oxidized is going to generate, and remember, during oxidation, electrons are removed. Those electrons are going to feed into the electron transport chain, which we'll talk about in detail in another lecture. And then via oxidative phosphorylation, generate a large amount of ATP. So you can think about catabolism in these 3 different stages. For the most part, we're going to talk about just stage 2 and stage 3 of catabolism. The other kind of soapbox that I like to get on is to remind people that catabolism is really not just about producing energy. You know, we always think of how it is that ATP is generated as we oxidize food. But catabolism does other things for us. It's really making 3 very critical commodities that bacterial cells and your cells need as well. Of course, one is energy. That's the easiest one to remember. But also reducing power. So you'll notice that for every one of these oxidation steps that occurs, like here, you're getting the generation of NADH. So you recognize that electron carrier as being reduced, having an extra electron, because it has a proton with it. Same thing with the TCA cycle. The TCA cycle produces a huge amount of reducing power. In other words, there's a lot of oxidation steps in here. So there are a lot of electron transporters being reduced. NADH, FADH2. These are all electrons looking for a place to go. Now, those electrons can be fed into energy production, such as the electron transport chain and the production of ATP. But those electrons have other very critical uses in the biology of the cell, in pathways that we won't talk about, the anabolic pathways. So many times when things are being made in a cell, such as amino acids or fatty acids or lipids, there are steps in the process to make those things that require a substrate to be reduced. And so the electrons that are being generated here in the TCA cycle are not all ending up in the electron transport chain, as you tend to think about, because you've all memorized the TCA cycle, you've all learned about the electron transport chain. So there's a tendency to think that all of the electrons that are generated in those oxidation steps end up going into the electron transport chain to generate ATP via that F0, F1 ATP synthase. They don't. Many of them end up being used for completely different purposes, such as the creation of nucleic acids, things that require reducing power. So reducing power is an absolutely critical commodity, not just for energy production, but for anabolic pathways. And then precursor molecules. So one of the underappreciated aspects of these pathways that you've all memorized numerous times is that not all of the citrate, for instance, that's synthesized in the TCA cycle goes on to isocitrate. Each one of these steps, or probably each one of these steps, has at some point a siphon coming off from it. There are other anabolic pathways. There are things to create amino acids. So I've given a couple examples that are shown in this figure. Alpha-ketoglutarate and oxaloacetate are actually going to be drawn off at some level from this pathway to synthesize amino acids. So not every carbon that enters here goes all the way around this circle or is generated in CO2. At some level, things are being siphoned off to do other things. So catabolism also generates a lot of precursor molecules, and this is a really important aspect of this consideration. Okay, so we're going to talk about glycolysis now, and I just want to make this blanket statement. I know that many of you have memorized this before. Some of you could probably come up and recite it. I do not ask for memorization. I don't want you to memorize this cycle or any other cycle that we're going to go over. So my philosophy is this. There is great value in knowing these pathways. So when you take a biochemistry class with Randy or Lisa or Doug or whoever else teaches biochemistry, there is great value in being able to know all of the enzymes, know all of the where the electrons are going, follow the electrons. I think that's wonderful. But what I want you to remember is this is not a biochemistry class, and I want you to kind of be able to view catabolism from 20,000 feet. I want you to be able to have an appreciation for how the parts fit together. And my worry is, and this was the case with me when I was taking biochemistry classes and such when I was in college, was that I lost the appreciation for how everything really worked because I was so caught up in trying to remember the order of the enzymes and how they were regulated and what bonds were being broken and things like that. So there's no memorization here. What I really am preaching here is appreciation for these pathways and how they work together. Hopefully, what this does is actually complement what you've already learned in biochemistry classes or what you will learn in biochemistry if you haven't already taken it. So, an appreciation for the beauty of how all of these parts fit together, and then that will complement your understanding when you actually go through and look at the regulation of hexokinase and what bonds are being broken and stuff. But really, in terms of this glycolytic pathway, what the Embden-Meyerhof Parnas pathway, as it's sometimes referred to, is probably the most common means, but not the only means, to break down to catabolize glucose in this second stage of catabolism. So glucose is entering here. Remember that when we talked about the phosphotransferase system, I mentioned that glucose is transported as glucose 6-phosphate. So that transported in glucose in that way fits in at this first stage. So as you look through here, it's not until you get down, so nowhere in the 6-carbon stage is there any energy production. As a matter of fact, there's actually a loss of energy. ATP is being extended to phosphorylate this molecule. So at the first stage, there's no energy yield, only energy input in this stage. So this is one of the themes that I like to point out. As important as glycolysis is, it's not that important in terms of the energy budget of a bacterial cell. It does, unless you're a streptococci that doesn't have the ability to do the TCA cycle or the electron transport chain, and you haven't evolved to be really champions at the glycolytic pathway, glycolysis is really not that great a mechanism to generate energy. And most of the sugars that bacteria use in the environment are actually not glucose to begin with, right? So most sugars in nature that bacteria are going to have access to are not 6-carbon hexoses, but they all have to be converted either to glucose or generally oftentimes to fructose so that they can feed into this glycolytic pathway. That's why I mentioned earlier that bacteria will import glucose to the exclusion of every other carbon source. If glucose is there, it's going to use it because it doesn't have to go through all of the enzymatic gymnastics to create something like fructose from it. So the 6-carbon stage is entirely preparatory. So all it's doing is setting the stage for what's occurring below this line. So there are no oxidation reduction reactions in this step or in this first stage. Now remember, I want you to, even though I don't want you to memorize this pathway, you do need to be able to recognize where oxidation and reduction steps. So anytime you see an oxidation of, in this case, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, you can recognize that as an oxidation because here's the electron carrier, nicotinamide adenine dinucleotide, NAD+, in the oxidized form, it becomes reduced by and glyceraldehyde 3-phosphate is oxidized. So here's the production of the first production of what's referred to as reducing power. Here's an electron on a carrier that can do some work, that can do some good, if the bacterium is equipped for it. So again, as you go further down here, now you begin to see the ATP is generated at 2 different steps here, but a relatively modest gain in energy in terms of ATP from the glycolytic pathway. But that's all occurring in that second stage. So these redox reactions that occur by the dehydrogenase enzyme generates that NADH, that reduced electron carrier. All of these ATPs that are generated are examples, again, just want to reiterate so you understand this term, by substrate level phosphorylation. The phosphate group that's put on here is coming from the substrate. So that ATP is being generated by substrate level phosphorylation. So the net energy yield is only 2 ATP, and pyruvate is the end product, right? But pyruvate is still a highly reduced molecule, so that means that it's still very energy rich. You can really think of that in a way as the end of the glycolytic pathway, and if we were going to talk about fermentation right now, fermentation would really be what is the fate of that pyruvate molecule. So we'll talk about the fact that many bacteria, if they can't do the TCA cycle, if they don't have electron transport chain, will use pyruvate basically as a waste product at that point. They will reduce it, they will excrete it as a waste product and lose a lot of energy. That is a very inefficient system. So as another pathway to consider when we're thinking about glycolysis is a pathway, again, no memorization here. What I want is an appreciation for this. This is the pentose phosphate pathway. This is a very important process in catabolism. I'm going to link it with catabolism, even though, so here, the entry point to the pentose phosphate pathway is glucose 6-phosphate. So recognize that as one of the early steps in the glycolytic pathway. This pathway is really crucial actually in anabolic pathways. Because as you look through this pathway, and again, do not remember what these molecules are, but remember that this 6-carbon sugar, glucose 6-phosphate, entering the pentose phosphate pathway, also known as the hexose monophosphate shunt, moves through this, and you get all types of different carbon skeletons created in this. Enzymes such as the important enzymes here are the transketolase and the transaldehyde enzymes. You'll notice if you look at these, you'll see 5-carbon molecules like ribose, 7-carbon molecules like cetoheptulose. These are molecules that are not involved in the energy production. If you look through this entire pathway, you do not see ATP generated anyplace. There is no direct energy produced here. What's instead produced are these very important precursor molecules for anabolic pathways. So when a bacterium has all of the energy that it needs, if it's fat, dumb, and happy, rather than run glucose 6-phosphate through glycolysis to generate more ATP, it can instead shunt that glucose 6-phosphate into this pentose phosphate pathway. Because what that's going to do is going to take that 6-carbon molecule and make things that can be used to make DNA and RNA. It's going to use things that can be made into fatty acids, into vitamins, into amino acids. So this is, again, an example of how catabolism is not simply important in generating energy, but it's also very important in generating precursor molecules. So all of the molecules that are listed in here are going to be sucked off into various anabolic pathways to make everything that the cell needs to be built. The other thing that's really critical about the pentose phosphate pathway and why we talk about it is that it's also one of the key areas for the generation of reducing power. Now, I don't know how well this shows. So here, you're seeing a different electron carrier. So in glycolysis, the electron carrier that facilitates oxidation of things like glyceraldehyde 3-phosphate is NAD+, nicotinamide adenine dinucleotide. The electron carrier that's used in the pentose phosphate pathway is related but different. It has a phosphoryl group. So NADP, nicotinamide adenine dinucleotide phosphate, is also, this is the oxidized form. It can become reduced during this oxidation reaction between glucose 6-phosphate and 6-phosphogluconate, and it generates NADPH, reducing power. NADPH has a very different fate than NADH. NADH is, for the most part, if it's going to be used for anything to generate more energy, it's going to donate those electrons into the electron transport chain, if it exists. So that electron carrier is much more dedicated towards generating energy. NADPH is the preferred form of electrons for anabolic pathways. So as pathways need electrons to help them synthesize things, more often than not, and this is not an all or nothing rule, but NADPH is much more frequently the source of those electrons. And so you see now in two really key examples of how the pentose phosphate pathway is really important in anabolism. It's generating reducing power for anabolic pathways, and it's generating the various carbon skeletons that are needed in anabolic pathways. And so bacteria, and your cells as well, have the ability to, when ATP is at high levels, they don't bother to take glucose 6-phosphate into the glycolytic pathway. They can shunt it and shunt more of it into the pentose phosphate pathway to allow the cells to grow. So again, this is just another way to look at this same pathway, the pentose phosphate pathway. And again, I got ahead of my bullets, but if you look through this pathway, again, just for an appreciation, what you appreciate, and I know you appreciate this, is that there are there's no usable energy being generated here. No ATP, no GTP, no NADH. So the things that would come from the glycolytic pathway or the TCA cycle. Instead, what's being produced here is the reducing power and the precursors. So this is very active under energy replete conditions, meaning energy is at high levels inside of that cell. I only put this one up because it kind of shows how these two pathways, the glycolytic pathway and the pentose phosphate pathway, are related to one another. So starting again with glucose, going to glucose 6-phosphate, if the cell is depleted of energy, then the pathway on the left, glycolysis, is going to be favored. Because even though there is only a net gain of 2 ATPs, if the cell really needs energy, this is the pathway that it needs to go into. But you can see that glucose 6-phosphate can be run through here. And actually, at various points, but especially here at the end, fructose 6-phosphate can be synthesized, so it can be fed back into the glycolytic pathway. So that's why it's called a hexose monophosphate shunt. It can shunt 6-carbon sugars into this pathway, and then eventually bring some of those carbons back into the pathway if necessary. So that's why it's called a shunt. So this figure is really only useful because it shows how these two systems interdigitate with one another. And so for just a minute to get back onto my soapbox about the metabolic diversity that microbes have that we don't have, the glycolytic pathway that you have all learned stands in every eukaryotic cell. It's the same for yeast as it is for you and I. And it's the same in many bacteria. Many bacteria use the virtually an identical Embden-Meyerhof-Parnas glycolytic pathway. But bacteria actually have other distinct glycolytic pathways that don't exist in eukaryotic cells. So here's one, a different one called the Entner-Duerdorf pathway. Again, no memorization involved here. My point is just to point out that glycolysis, as you've learned it, is not the only glycolytic pathway in the world. The Entner-Duerdorf pathway is featured in a large number of bacteria, many of which are known to you. many of which you will actually assay for products from in the laboratory class. It's a distinct form of glycolysis, oftentimes found mostly in gram negatives. It actually has some very important economic, at least historically, it has some very important economic effects. So there's one bacterium that falls into the group of pseudomonads that occurs naturally on the plants of a magui cactus in Mexico. It's just part of the normal flora. It doesn't hurt the cactus, it doesn't help the cactus. But people noticed that when they would produce originally cactus beer or pulque by crushing the leaves, or perhaps it's the root, I'm not sure which one, of the magui cactus, that fermentation was very different. It's not the type of fermentation that you and I think of in beer and wine. That's done today exclusively by yeast. Traditional cactus beer, which ends up eventually being, one of my favorite drinks, tequila, that fermentation, at least traditionally, and I think still in some small distilleries today in Mexico, is done by bacteria, not by yeast. And this Zymomonas mobilis is a bacterium that doesn't use the typical glycolytic pathway that you and I use. It uses this Entner-Duerdorf pathway. So I don't know whether the unique flavor or the unique effects that tequila has have anything to do with the Entner-Duerdorf pathway or this bacterium, but I like to think that part of the reason why tequila is that special kind of liquor, and I think many of you know what I mean by that special kind of liquor, I think it maybe has to do with the microbial metabolism. So, as I said, I don't want you to remember a damn thing about the Entner-Duerdorf pathway, except that it's a glycolytic pathway, but I want you to remember how tequila is traditionally made. That's the important lesson. And then this is just another chart to give you kind of an appreciation for the fact that not only do bacteria have alternative glycolytic pathways, they sometimes can use multiple glycolytic pathways in different conditions. Bacillus subtilis, the bacterium that you did spore stains on this year, for the most part prefers to use an Embden-Meyerhof pathway, but it has a very distinct pathway as well. It has this heterolactic pathway that we're not going to talk about because I don't understand a damn thing about it, but it's a different glycolytic pathway. But there are some really important human pathogens like Pseudomonas aeruginosa, which we'll talk about later, which is important in burn patients, in cancer patients, in cystic fibrosis patients. That's a bacterium that uses a very different glycolytic pathway than you and I. It uses the Entner-Duerdorf pathway. And the bacterium that causes cholera actually is one of those bacteria that has both pathways. It prefers to use the Entner-Duerdorf pathway, but under certain environmental conditions, it will switch over and use the same pathway, largely the same pathway that you and I use. So again, metabolic diversity in bacteria is really rampant. So the last thing I want to talk about today, and I'll continue this in the screencast that you'll view later, is talking about fermentations. So for you and I, we think about fermentations as something that's useful for us. Not only just beer and wine, but I mean, we depend on fermentation for things like sauerkraut and Swiss cheese and a lot of different food products as well as alcohol products. So that's what fermentations do for us. But what do they do for the microbe? Because the microbe, using the glycolytic pathway, faces a serious problem in using the glycolytic pathway. And that's because these nicotinamide adenine dinucleotide pools are very small. Any individual cell doesn't have a lot, doesn't have an endless supply of these electron transporters. And so it doesn't take very long for the NAD pool to be depleted during oxidation in glycolysis to turn all of this NAD into NADH. If that happens, if all of this NAD is turned to NADH, oxidation stops. Glycolysis stops. Energy production all depends on glycolysis to get things rolling. And if that oxidation stops because there's no electron carrier, because they're all reduced into NADH, that's a major problem. This is what fermentation does for the microbe. What happens in the solution here that bacteria have used in terms of being very efficient at fermentation is that they regenerate the oxidized form. They take away that electron from NADH by oxidizing pyruvate. So I mentioned that really the end of the glycolytic pathway is this molecule pyruvate, which is still highly reduced, still very energy rich. Many bacteria, like the streptococci in our mouths and throats right now, at the end of glycolysis, will now use that pyruvate in a fermentation reaction where pyruvate becomes the internal electron acceptor, the final resting place of the electron. Pyruvate, in this particular case, substitutes for the oxygen that you and I are using, the final resting place of that electron. So what happens is that NADH dumps its electron onto pyruvate, reducing it to lactic acid, a waste product. That's what's secreted by streptococci. That's why streptococci are very good at producing acid, which can decay teeth, that can break down the enamel of teeth, is because they produce a lot of lactic acid. This is still a very good carbon and energy source, and other microbes will use it. But what this does for the bacterium is it regenerates this NAD pool, so it allows glycolysis to continue. So even though glycolysis is not incredibly efficient at generating ATP, bacteria that use only the glycolytic pathway, like the streptococci, regenerate their NAD pool by simply reducing pyruvate to something like lactic acid. There are other end products to fermentation, luckily for us, one of which is alcohol. But these reduced pyruvate products are microbial waste products, so they're secreted. They end up being food for other microbes. They end up supporting some very elaborate microbial ecosystems because there are microbes that will use the fermented waste products of bacteria. We'll talk about this when we talk about archaea, because in ecosystems, in wetland ecosystems and marine ecosystems, these fermentation products of bacteria are actually inhibitory to the bacterium. So if lactic acid builds up to too high a level, the bacterium that produced that waste product will now be killed. But the archaea will use these products, these what are called short-chain fatty acids, to produce methane, to get rid of that lactic acid. So lactic acid bacteria often are using only glycolysis as their energy source. And so that waste product ends up causing dental caries. Now, part of the problem for bacteria like streptococci is that although they've become very highly evolved, very adept at doing the glycolytic pathway to generate energy, they don't have complete TCA cycles. So what that means for the bacterium is that it can't produce a lot of the precursors that it needs to synthesize amino acids and fatty acids. So bacteria like streptococci that don't have a glycolytic pathway have very complex nutritional needs. When we grow them in the laboratory, we have to baby them. We have to feed them practically everything because they can't synthesize a lot of their own amino acids. They can't synthesize a lot of their nucleic acids. So we grow them in very rich media in the laboratory because they need all these vitamins and growth factors that they can't make because they don't have the catabolic pathways that make the precursors for these things. And as I already mentioned, the lactic acid bacteria are important in yogurt, sauerkraut production, all of these kind of foods that we like to eat. And I think I'm going to take care of the rest of fermentation and such on the screencast. Have a great weekend, and I'll see everybody on Wednesday.